Well, good morning, everyone, and thank you for joining me today in this fifth webinar in the ACEL webinar series. My name is Josephine Lembong. I am the Senior Manager of Science and Industry Affairs at the Alliance for Regenerative Medicine. Today's webinar is on the topic of ancillary materials and will be delivered by the lead contributors of Chapter 6 of ACEL, who I will introduce in just a few minutes. I think everyone here might be familiar with ARM, but for those who are not, we are the leading international advocacy organization championing the benefits of engineered cell therapies and genetic medicines for patients, healthcare systems, and society. As a community, ARM builds the future of medicine by convening the sector, facilitating influential exchanges on policies and practices, and advancing the narrative with data and analysis. We actively engage key stakeholders to enable the development of advanced therapies and to modernize healthcare systems so that patients benefit from durable and potentially curative treatments. We represent more than 500 members across 25 countries, including emerging and established biotechnology companies, academic and medical research institutions, as well as patient organizations. As I'm sure everyone here knows, manufacturing for cell and gene therapy is a complex process and is often a rate-limiting step in the commercialization of regenerative medicines. The industry as a whole is in the process of rapidly scaling up, and there are more phase 3 studies and more commercial product approvals that are anticipated. However, products are being held up at the BLA stage due to CMC issues. We constantly see that advances are being made in the manufacturing technologies, and the experience in the workforce are overall improving, which expands the process understanding. However, standards and best practices in the field are not advancing as quickly, and this is the pre-competitive area where ARM focuses our efforts in to further advance the process understanding in the industry to facilitate product development and commercialization. ACEL is a project that ARM undertook starting several years ago after feedback from our members about the CMC issues that the field as a whole is experiencing. It is a best practices document which incorporates collaborative case study presenting the implementation of quality by design principles in cell-based therapies. And to make ACEL an effective resource and reflective of the ongoing innovation, we chose to focus on immune cell therapy and in particular an autologous CAR T therapy case study. These efforts follow the success of AGENE, the sister project of ACEL, which focused on AAV vectors, as well as AMAP, which was instrumental in allowing advances to occur in the monoclonal antibody industry in terms of improving manufacturing quality, optimization, and not to mention the driving down COGS. We at ARM are very fortunate to have a large number of subject matter experts from our member companies. There are many contributors that are involved in ACEL, and we're also very grateful to NIMBLE, who generously provided the funding, which allowed us to employ technical writers, without which we wouldn't be able to complete this project. The document is intended to benefit the entire field, so it is available as open access in our website at this link over here. This is what the contents of the documents look like. As you can see, it covers quite a big scope. Chapter 2 covers regulatory considerations, comparability and standards, and then 3 and 4 are really the foundational principles of QBD. 5 and 6 touches on the starting materials and ancillary materials. 8 details the manufacturing process, and then analytics, control strategy, and last but not least, facility design. Today's talk will cover the ancillary materials principles and case studies laid out in Chapter 6. The webinar today is the 5th out of a 9-part webinar series for the ACEL educational rollout. The schedules for the webinars will be posted in this link. Our hope is that ACEL, both the document and the webinar series, can serve as an educational tool to the community. We hope that this document will be used by product developers, educators at universities, and we also have the aspirational goal that this will be used to help educate regulators. Before we move on, I just have one housekeeping item. Please use the Q&A function to ask questions, and we'll try to answer as many as we can at the end of the talk. If we didn't get to all of your questions, we will compile them and post a response in the ACEL webinar website. And without further ado, I would like to introduce our expert speakers for today's webinars. Dr. Ezequiel Zylberberg is a Vice President of Product Development and Planning at Akron Biotech, a manufacturer of GMP-compliant materials and services to enable cell and gene therapy development. He has spent nearly a decade in the cell and gene therapy industry. In years prior, he worked as a strategy consultant across an array of technology-intensive sectors, including semiconductors, aerospace, and biopharma. He completed his doctoral and postdoctoral training in management and economics. And our second speaker today is Dr. Ramon Montero. Dr. Ramon Montero is an award-winning and highly published senior scientist with over 10 years of academic and private sector experience in biomedical engineering and rapid prototyping for research and development. Ramon is a motivated and results-driven professional with an aptitude for research, analysis, and creative problem-solving, with a reputation for being a communicative and personable professional with the capacity to lead complex projects by employing innovative methods and programs within collaborative laboratory environments. Ramon and Ezequiel, thank you again for taking the time out of your busy days to be here today, and I will turn it over to you to start your presentation.
Thank you, Josephine. Okay.
All right. So we ready? Awesome. Great. So first of all, thank you so much to ARM for providing us this opportunity to really, dig deep into this issue or this field of ancillary materials. So thank you very much for that. And I hope everybody's having a great morning. So again, today we'll just be discussing the ACell book chapter regarding ancillary materials and the importance of understanding their quality and suitability for use, specifically in the manufacturing of cell and gene therapies. So our goal is to detangle and bring a bit of clarity to this highly complex gray area of ancillary materials and the regulatory bodies that influence them. Additionally, we will introduce some risk mitigation strategies as suggested by different guidance documents from different jurisdictions and regulatory authorities. My colleague Ezequiel Zylberberg will dive into concrete observations and examples that we have come across as a company in this complex field. We can go to the next slide, please. Awesome. So first, we really need to define exactly what an ancillary material is. And I just want to make sure we understand that in this globalized supply chain, the definition and terminologies can change and be a bit misconstrued. Especially in Europe, sometimes these are referred to as raw materials, which ancillary materials does fit in the more broader umbrella of a raw material, but it does drive a more specific bit of a definition there, which is it's an ancillary material is intended for it's a material used and it's intended in the manufacturing to be used in the manufacturing process, but it's not intended to be part of the finished cell or gene therapy. So some examples, right? Some of the usual culprits for ancillary materials are monoclonal antibodies, cytokines and growth factors, serums and enzymes, and substrates for scaffolds, right? So in addition to this, and kind of like a side leg to that, some of these consumables, such as biobags and tubing, which come in direct contact with the final cell and gene therapy, but clearly are not intended to be administered to the patient. So these are just some of the more common examples, but there could be more that can be identified as ancillary materials. So the ancillary material complexity, right, leads to a difficulty in assessing the risk, and we really understand that at the end of the day, the cell and gene therapy manufacturers are working with the hardest of all raw materials, which is the cells from the patients themselves. So we understand that you guys rely a little bit on these kind of labels and in these terminologies. So because these materials are not well defined in the regulatory space, and the burden ultimately lies on the cell and gene therapy manufacturers, many of you have to rely on this gray area of, grades, different use statements, as well as CNO risks profiles. So while these labels can shine some light and facilitate their selection, ultimately it's required by the cell and gene therapy manufacturer to really understand their usability within the intended process. So again, many of these don't have an official legal definition. They're used conventionally across the industry to mean different things, but ultimately the user needs to really dig and understand the critical quality features of the materials that may impact the final product. In absence of these of regulatory oversight of these materials, users need to utilize a risk management strategy to determine suitability. The landscape of regulatory bodies add yet another layer of complexity based on geographical location of manufacturing and or sourcing of these materials. So there are different jurisdictions and therefore different regulatory authorities that govern them. Some of these international agencies or organizations, such as the World Health Organization, the International Organization of Standardization, ISO, and the International Council for Harmonization, they lay out a risk management framework and try to align the different regulatory agencies to facilitate the process and further enhance the chances for success of the finished therapy. So again, these international organizations attempt to give some sort of cohesiveness within these health organizations or across the different health organizations. And the regulators in each target market have slightly unique requirements and specifications for final approved cell therapies. And clearly there's overlap, overlapping principles, but the slight differences between the core regulatory agencies, again, adds that level of complexity which has to be considered by the developers and to which the ancillary material manufacturers should strive to be aligned with. So given the globalization of supply chains, ancillary material manufacturers need to be knowledgeable or at least aware of the specific differences between the core regulators. Something that can help bridge the gap between cell therapy manufacturers and ancillary material manufacturers is when the ancillary material supplier manufacturer can communicate directly with some of these major regulatory agencies, and they could do so via U.S. master files, EU substance master files, and PDMA from Japan. Although there are clear nuances into the ways in which these approaches can be different. But anyway, what this allows is to kind of remove that the cell and gene therapy manufacturer from playing a middleman, and they don't have to dive into the complexity, or the specifics of the ancillary material manufacturing process itself. So this helps and gives a high level of confidence on the material being used for the finished therapy. So that said, basically AM suppliers and manufacturers are doing or should do, right? And that's what Akron definitely strives for, to do a little bit more of the legwork to ensure success of the finished therapy, which quite frankly is just mutually beneficial for the success of both companies, right? The supplier, a manufacturer of ancillary materials, and the cell therapy manufacturer. So here, right, some of the more relevant ICH guidelines for key AM considerations, right, as stated by the ICH, would be sterility, stability, virus validation, and then cell lines. And I want to kind of park here for a second with the cell lines because, for example, CHO cells and others are animal-derived cell lines and are cause for more stringent considerations by regulatory authorities. So while these are principles implemented for pharmaceutical products, If we use them as guidelines, it facilitates the progression through their approval process. So as you well know, regulatory authorities want to know all the details of these products and materials used in the manufacturing process, albeit from an ancillary material standpoint, they are not present in the final product. So nonetheless, this is not intended, obviously, to replace due diligence required ultimately by the cell therapy manufacturer to ensure efficacy and safety of the materials and their impact thereof on the finished therapy. We can go to the next slide. Right, so kind of to preface this, right, for Akron, at least, our target space and commitment to our client is to adopt and follow a stringent QMSs, right? So within USP 1043, there's a definition in tiers, right? So there's like levels. There's 4 tiers for the use of ancillary materials, right? To define the risk of use of ancillary materials. So the first tier would be basically an already accessible or in the market, right, pharmaceutical product. And that comes with its own caveats, right? So cost, presentations or format, as well as maybe concentration may not be ideal. So for a cell therapy manufacturer to take the approach and say, okay, we're just going to go with all tier 1s, it may not be cost effective, and honestly, it may not be from a manufacturing standpoint the right thing to do because of these little caveats, right? So tier 2 is kind of the space where Akron likes to live in, and as a company, we really strive to be in this tier, which is a bit of a sweet spot where we have a high quality product without the cost of a pharmaceutical product, and this ultimately facilitates the procurement of the material, not only from a cost standpoint, but also from, not requiring any special pharmaceutical licensing or anything like that, or license or anything like that. Tier 3 and tier 4 are a little bit the riskier ones, clearly. These are less well documented type of materials. And tier 4 adds, that whole level of maybe, harmful impurities and may contain animal or human derived products, which have obviously the ability to harbor advantageous agents. So as mentioned in the previous slide, these tiers, specifically 1 and 2, rely on a good quality management system, right? So some of the more general ones, or the ones that we should follow, is ISO 9001, which provides an exemplar QMS that ancillary material manufacturers should adhere to and can be certified against. 13485, it's a more stringent one. Is better aligned with this field, and frankly considered a higher level of quality compliance to which Akron is certified against. And lastly, as of 2018, there's ISO 2399, which provides a risk management strategy, right? Specific for the ancillary material use, specific to cell processing of cellular therapeutic products, right? So Akron considers this strategy I outline in this ISO chapter when manufacturing ancillary materials used in the cell and gene therapy space. So this sets up, right? All of this that I just mentioned kind of sets up a lens through which we can now view some of the examples and observations that my colleague Ezequiel Zylberberg will be presenting.
Thanks, Ramon. And in a way, I think what's important to do, and what we want to do here is kind of bring this down to a level where we can all understand it. I would assume that a lot of folks on this call think a great deal about ancillary material quality, sourcing, risk management. And given that we've been in this space for quite some time in terms of supporting cell and gene therapy production with GMP compliant materials, we've really kind of what we really wanted to do was provide folks a series of best practices or a series of observations, things that we've noticed are quite common when folks, especially in the early stages, are sourcing some of these materials and qualifying them as a means of really complicating this category of product, right? I think we tend to think about these as kind of pieces of a puzzle, right? Because manufacturing cell therapy is quite complex. But I think it's also important to understand that each of these building blocks has an inordinate degree of complexity within it. And so understanding that complexity and unpacking it allows us to better assess and mitigate the risk that can be presented, right? So I think that the three main observations that really come to mind here are, one, oftentimes we don't consider the risk that animal or human-derived materials might introduce into the product. And while it might be a consideration, it's not assessed to the degree to which is required. Ultimately, what we see all too often is questions directly from regulators in response to an IND filing, for example, where questions are brought up around the suitability of certain materials. Folks will come to us and say, hey, do you have a product that meets these standards? So you shouldn't be finding out for the first time that you have issues around human or animal-derived material sourcing as a response from the FDA, right? Think about these things earlier on, and we'll go through a couple case studies on these. Two is really variability in cytokine activity and mass reporting. We find that this is actually quite common and it's natural. These are biologics in their own right. So again, they have their own degree of complexity, and ensuring interoperability between suppliers is not straightforward. It's actually quite challenging. But it's really an important Rubicon we have to cross as an industry if we're really going to get to a modular supply chain that can support commercial level production. And three is really the robustness of the supply chain and commercial readiness. Oftentimes I think it's quite common in the early stages, right? We need to get some clinical data because that's going to allow us to unlock the next tranche of funding. And so a lot of these problems are kind of punted forward, so to speak. But in doing that, we lose a critical opportunity to mitigate long-term risk. And so building in supply chain robustness from the very beginning is actually quite important. So to dive in a little bit on the first observation, I really want to bring to the fore two case studies of folks that came Akron's way with different but related problems. One was a clinical stage NK cell therapy company in Europe. They'd gotten feedback regarding their current supply of human AB serum and virus and activation thereof, and they were experiencing quite a bit of variability with off-the-clot AB. And so they were looking for a more reliable source. And then the second case, really a BLA-enabling T-cell company that had really complicated requirements that many of you may be aware of in certain jurisdictions in Europe, I think especially in France, but elsewhere as well, around needing small pools of donors to mitigate the risk of adventitious agents being introduced into the cell therapy product, given that the cell therapy product can't undergo a process of terminal sterilization. So really, this was not a viable commercial strategy, right? You can't build a commercial product if one of your core materials is limited to 17 donors, where each process might require a substantial amount of that. So how do we think about viral safety in general and viral clearance, and how you mitigate the risk that these pathogens can introduce, right? So to me, it starts with donor screening, testing of these materials, whether it's nucleic acid testing or serological testing, making sure that all kind of donor material is tested in accordance with 21 CFR 61040, that all the virus tests are conducted appropriately, and that viral testing is conducted not just at the donor level, but throughout the manufacturing process as well. And beyond the testing, it's really viral clearance, and that at the end of the day becomes one of the more critical items to consider, right? Because regardless of how effective your screening mechanisms might be, and regardless of your alignment with the relevant CFR, there's always the risk of introducing adventitious agents in these processes, right? These are plasma-based products. And so you need to have a viable viral clearance strategy. So what we have on the right is really a variety of ways to think about this, right? Heat activation is more appropriate for complement proteins than it is viral clearance, but you can think about solvent detergent treatment. You can think about gamma radiation. There are a variety of means of achieving the log reductions in viral load that are going to be relevant and sought after by regulators, right? And so we really seek to build these into our products. And the second one is really thinking a lot about the risk that animal-derived components can present to a process and how to properly classify them. And by classifying them, really assigning an appropriate degree of risk that they introduce and provide a means of mitigating that risk, right? So it really depends on the level at which these materials are used, right? Is an animal-derived component used as a primary material to produce the final ancillary material, right? It's a bit of a further degree of separation from the cell therapy product, but there's a non-negligible degree of risk that you have to contend with at the primary level. Is it at the secondary level? Is it something that's used to produce primary materials that then go into the ancillary material that goes into your cell therapy process? Each of these tiers carries its own degree of risk, and you need to make sure that BSE risk is classified by country of origin. The IOE terrestrial code will help to assess degree of risk, and the specific tissue source. What animal tissue does this product come from, because that can confer a greater or lower degree of risk of BSE onto the product, right? So any supplier BSE TSC statement needs to outline not just the source of the components in terms of the tissue, but also the country of origin and the processing level, because ultimately all three variables are critical to assessing risk. And just to kind of give you an idea of how we've thought about addressing the case studies that were presented early on, what we did was we actually partnered with Octapharma, right? So this is Octapharma manufactures a solvent detergent treated human plasma product, has a massive donor network that it leverages to source the plasma. And in really driving a robust viral clearance process, primarily on the basis of solvent detergent treatment, it's actually able to achieve a significant log reduction across a variety of transfusion relevant viruses, which overcame a lot of the issues that regulators in certain European jurisdictions had around maintaining large donor pools. So what we've been able to do here is maintain donor pools that go from 600 to 1,500 donors, but at the same time provide a robust enough viral clearance strategy to enable the use of this product in cell therapy manufacturing, in addition to testing, right? So you're able to overcome a lot of the lot-to-lot consistency issues by pooling larger batches. You're also able to overcome a lot of the safety issues associated with large donor pools by implementing quite a robust viral clearance process. So just in sum on the viral clearance and on risk associated with human and animal ancillary materials, you have to anticipate that these materials are going to experience increased scrutiny over time. The bar is not getting any lower for these materials, right? Over time, it's getting higher, and the degree of scrutiny that regulators are going to place on materials that are incorporated into the cell therapy manufacturing process is going to be higher. So you really have to make sure that the product does meet all donor screening and viral testing requirements. Secondly, you really need to make sure that it meets all the viral clearance requirements as well, and that it's going to be a suitable material. Again, you shouldn't be receiving feedback on your IND regarding the suitability of a material. You really have to consider upfront how much risk these products can introduce and how you're going to manage that risk. And lastly, lot-to-lot consistency. We didn't talk about it, but it's quite a common practice, and we've seen it time and time again, cherry picking lots of AB serum. And while it might be suitable as a near-term strategy, it's not a means of building a robust commercial supply chain, right? You can't rely on a certain lot of material being available. You have to be able to move to something that is ubiquitously consistent, that is consistent from batch to batch. The second observation is really around how we measure, assess, and report activity and mass for cytokines. And this is really important because variability on this front can make it really complicated to qualify a secondary source, and it can be really complicated to maintain the consistency of your own manufacturing process, right? And I'll start with a case where, we had a customer that was using interleukin-12 in their process, and for regulatory reasons was not able to move forward with its current source of interleukin-12 and was looking for a new primary source. Now, what they did is basically assessed both materials certificates of analysis, looked at the specific activity listed, and found that just based on analyzing specific activity, one would need 24 times the amount of Akron material to achieve the same outcome as their existing vendor. And this was quite puzzling to us, in part because the specific activity of a given cytokine is largely intrinsic to the cytokine. It varies relatively little and shouldn't oscillate in this to this extent, right? And so what we did was we evaluated these cytokines side by side, and we wanted to really understand, is this really a difference in activity, or is it a difference in the means that activity is tested and reported? And so what we found actually in evaluating confirmed our hypothesis that in reality, these are both quite similar proteins, and the specific activity should therefore be quite similar. We don't have any post-translational modifications on either. There are no meaningful differences that should confer different degrees of activity. This is really about just using a different cell line and assessing biological activity in different ways. So what this allowed the folks to do is really have a viable crosswalk between their suppliers to ensure that their process would not be impacted by a change, right, from an operational standpoint. And the way we generally think about biological activity assays, executing them, analyzing the data, and reporting is via what we call parallel line analysis. So we run parallel line assays evaluating the parallel line regions of the sigmoid curves to ensure that we're really capturing statistically significant results when we look at the information and reproducible results from batch to batch. So we follow USP 1032 and how we design these biological activity assays, and USP 1034 and how we analyze these assays. And this is really important because the way that biological activity is assessed and analyzed can have an outsized impact on how these proteins are used and the impact on the process from a user standpoint. So relying on an ED50 confers its own variability just purely from an analytical standpoint. So by looking at the parallel line regions of the curves, we're able to achieve a statistically significant outcome in terms of the data that we're reporting on these activity assays. And it's also important to note kind of a slight difference in nomenclature, but it can lead to important differences in the way that folks use cytokines, right? So a specific activity is going to be expressed on the basis of international units per milligram. And as I mentioned, a specific activity is largely going to be an intrinsic property of the molecule, whereas biological activity is generally expressed in units per milliliter or units per vial, and is going to vary based on the concentration of the product, the size of the aliquot. So it's important to kind of distinguish between these two when an assessment on dosing is being made. Now, the reason it's important is that ultimately your analytical technique in assessing biological activity, coupled with the robustness of the manufacturing process for these cytokines, in addition to the qualification and suitability of raw materials used in cytokine production, can all lead to either lot-to-lot consistency or inconsistencies from batch to batch. And again, this is another case where we saw significant differences from one lot to the next across different vendors. And again, some of it is associated with analytical techniques, some of it with validation of manufacturing process or lack thereof, and some of it is associated with raw material selection. So it goes back to saying these cell therapies are incredibly complex, and often we think about kind of the complexity of ancillary materials as exogenous or otherwise unimportant, and you know, we can basically rely on a certificate of analysis from a supplier or on a label to assess how we're going to use it, but it's just understanding that even that material is a biologic in its own right and confers its own degree of complexity, and you have to manage that degree of complexity. Otherwise, you're going to have differences in your process and lot-to-lot inconsistency that is difficult to explain unless you've been able to truly understand what the root cause of some of that inconsistency might be. And the last one on the cytokine reporting front, analytical front, relates to mass, right? So there are also proteins where, you know, the use has not been protocolized or defined, or let's just say it has not kind of entered, it has not become common enough to where its use justifies transition to international units for dosing. And folks really use mass, whether it's milligrams per ml or nanograms per ml. And this was a case where, again, we were looking at interleukin-7 and interleukin-15, and a partner of ours was seeing significant variability across several vendors in really the yields of their process associated with different vendors. And again, you know, it was a bit perplexing in that, again, we assume that the specific activities of these proteins are going to be largely similar. So what could be driving a lot of that variability? So what we actually did was kind of worked backwards, and we started with mass, right? If you're dosing on the basis of mass, then let's understand if the dosing is happening in a consistent way, right? So you look at what the mass is on the vial label, you look at what the mass is on the certificate of analysis where it's available, and then you run your own empirical assessment of what the mass might be. In our case, we did reverse HPLC to understand how much mass was in each vial. So we established that, okay, the mass on the label may not always be the mass that's in the vial. And there actually, in some cases, might be significant differences depending on what the specification is for mass, right? So you can't always look at the vial in terms of how much mass is in the vial and dose on the basis of that, because there might be some variability there. And I think we have to get to a place as an industry where we're able to standardize some of these assessments and move towards having mass reported out on a lot specific level. And what we actually were able to assess empirically on the basis of this experimentally defined mass and on the basis of running our own validated CTLL2 line biological activity assay on these three proteins was that, as we expected, the specific activity is quite similar, right? You're going to see differences associated with the cell line that's employed, or perhaps in the way that the analytical technique is developed and run, but ultimately you should see fairly consistent results from batch to batch. So what I think here is we need to really think a lot about standardization in terms of cytokine activity, the evaluation of cytokine activity, the evaluation of mass, because unless we're able to do that, we're not going to achieve a fully interchangeable supply base. And of course, you know, if we're not able to get to a place where we have interoperability between suppliers, we're always going to see variability in these processes, and it's going to be really difficult to scale a lot of these manufacturing processes. So it's really important to understand lot-to-lot consistency. It's important to understand the impact of variability across lots on your own process and what the tolerances really are around cytokine dosing, so you can understand how your CPPs impact your CQAs ultimately. And really what we've seen as best practices is folks that really go out and do a fairly extensive characterization of these biologics, look across several suppliers, rely on a third party, establish your own crosswalks, leveraging an international standard, as you know, those put forth by NIBSC or USP, to really assess, okay, what is my conversion factor going to be if I have to move from one supplier to the next? Can I establish that these are truly interchangeable? It's all part of establishing a commercial ready supply base, which is where we're going next. So Ramon, if you want to jump in here.
Yeah, absolutely. Yeah, so but just what Ezequiel said, the supply chain robustness is very important. And I don't want to go down these and just kind of read all these bullet points, but basically the gist here is, you know, kind of kick the tires, so to speak, right? Like, do you know, check those boxes, understand how, when moving from one stage to the other, can impact your final product, right? So we're gonna give an example of how this materialized, but basically I just want to give kind of a recent experience that I had, and I think the gentleman was doing the right thing. You know, he comes up to me, and just to kind of make everybody understand and have this very clear, I'm director of research and development, right? So he comes to me and he says, hey, is your RUO product going to cost me issues when I go to CGMP, right? So understanding that, yes, and some of the burden has to be taken up by us, the ancillary material provider, to kind of give that sense of stability and connectivity between one grade and the next, that is not going to just discombobulate your entire process. So here's an example of how that materialized with a project that I worked very closely with for substrates. This was an electrospun scaffold comprised of polycaprolactone. So the client decided to start with research grade material. And once we had all the bells and whistles kind of stipulated on the entire process was you know, determined, we decided to jump into a GMP grade material. And this is a very unique case, and the reason why we're presenting it is because that was the only factor that changed, right? So this is a direct effect of just changing from a research use only or research grade material into a GMP material. And as you can see, these are SEM images, scanning electron microscope images. Yes, there are a little bit of different scales, but there's a clear difference in which the research grade material performed as we wanted, and it yielded the within the specifications of the client, the material, you know, within the specifications of the client. And when we switch over to the GMP grade polycaprolactone, the process was non-viable, right? The yielded material was just so out of spec that completely useless, unusable. And this really obligated us to look at ourselves and say, okay, are we going to start over? And if we are, should we start over with a GMP material? And then that further kind of brought up the question of what about a secondary supplier? So if we're seeing this from a singular supplier between grades, what can we expect, you know, across suppliers? So intra versus inter supplier differences.
Now, I just want to kind of go in a different direction in a way, but it kind of is still tied into questions around kind of supply chain integrity and how you maintain a commercial ready supply chain, and kind of walk you through another case, right? So there's the sorts of cases where Ramon mentions, right? You're moving from an RUO material to a GMP, and there are massive differences in performance. Differences that can't be explained away, and that frankly can often lead back to bridging studies, additional raw material qualification studies. So much like that case where you really have to think about the fluidity of the path from RUO to GMP, you also have to think about it in terms of suitability of the product format and sizes, right? And in this case, we have a customer that really started with us in very early clinical stages, introduced our interleukin-2 100 microgram GMP product. So it was always GMP, but over time, as they kind of moved into larger batches of media, really had to scale up. Now, for us, the manufacture of these proteins, at least at the API level, is identical, right? So how we make these recombinant proteins is the same regardless of the aliquot. What differs is the fill finish process. So ultimately, the performance of the 100 microgram and the 1 milligram aliquot that they scaled up into was the same, right? It just allowed them to have to open fewer vials, frankly, and reconstitute less material to be able to manufacture larger batches of media. And lastly, this customer has since gone commercial and is switching over into bags, right? Because it enables direct sterile welding and integration of liquid cytokines into a manufacturing process, avoiding reconstitution and any open processing steps. So again, being able to move within the same product line with the same performance from small scale to large scale to closed system enables that transition from preclinical all the way out to commercial. And also just to say, you know, we at this point have worked with hundreds of cell and gene therapy companies in this space. And I think it's useful to reflect back that a lot of people talk about supply chain resilience, supply chain integrity, but the degree to which folks have actually qualified secondary vendors, gone and done technical visits, quality audits, done kind of material evaluations, and at least some degree of characterization work is limited, right? And this is totally understandable, right? There's lack of bandwidth. You're focused on the main problem, which is producing a viable cell or gene therapy product, right? You know, you can't chase every rabbit hole in terms of your supply chain. And it's relatively low on the list of priorities. And sometimes there's a fear of introducing changes to the process that you're going to have to explain, identify, and do an entire root cause analysis on later. So a lot of the times this is kicked down the road. But I think what we're saying here is by doing this early and by qualifying and integrating a secondary source early, you actually gain quite a bit of flexibility in that you have optionality if something happens. And we all experienced COVID, we all experienced the supply chain shortages. But also, you know, as you go commercial, it allows you to have flexibility and put downward pressure on COGS, because ultimately you have options, and allows you to maintain the continuity of operations during critical junctures in your company. You don't want to be held hostage, basically. And I think that more and more folks on the investment side need to really think about not just, do you have an interesting target? Do you have some interesting data? Do you have kind of an interesting position insofar as IP, but is your supply chain well designed enough to really withstand a lot of the complexities that are going to come your way later on, right? It's not going to be the top of the list, but it should be an issue that merits some degree of consideration. So just to summarize, we've gone through really 3 classes of observations that all tie back into Ramon's discussion in the ACEL chapter more generally on ancillary material quality, understanding it, assessing it, mitigating risk associated with it. And for us, these 3 buckets really revolve around kind of understanding, characterizing human or animal-derived materials, right? Everyone wants to go serum-free. At some point, it may be a reality, and I don't think we're quite there yet. So until then, we really have to mitigate risk, right? 2 is variability in terms of how we understand our cytokines and the recombinant proteins that are absolutely critical in terms of activation or differentiation of our cells, right? We need to achieve a greater degree of interoperability between suppliers and consistency from batch to batch so that we can indeed make this an exogenous variable that has no impact on your CPPs and ultimately your CQAs. And lastly, supply chain robustness. How well designed is your supply chain? Are you ready for commercial? And if you're not ready, have you at least kicked the tires, and do you have a good story for how you will be? So these are really, I think, critical considerations. So again, our view is focusing on safety. Ultimately, that's going to be what brings you down if you don't consider it in detail early enough and have suppliers that can really understand and assess risk alongside you. 2 is prioritize consistency. Right, you can't operate a process at commercial scale that is going to yield such variability that regulatory agencies are going to ask how much control you truly have over your process. And 3, future-proof your supply chain. It might seem like an onerous exercise, but I think a lot of it can be done on a paper-based approach, and you should be able to leverage your suppliers. And lastly, and this is something that we need to do better as a supply base, and that's Akron, as everyone that provides ancillary materials in this market. We need to do a better job of banding together and creating clear and consistent guidelines and crosswalks to be able to enable our customers to leverage our products more effectively, right? I've spent a lot of time in much more mature industries like semiconductor production, aerospace, and obviously they are enormously different from cell and gene, right? But a lot of the principles around supply chain modularity, around how you kind of maintain standards and clear interfaces between suppliers and developers are really important to prioritize, because ultimately it's going to be what allows us to create a robust industry. So that's really on us. And Akron, alongside a lot of our colleagues, are working with groups like the Standards Coordinating Body, like ARM, to start operationalizing some of this. How do we actually get to consistent guidelines around certificates of analysis, around analytical techniques, especially those that are non-compendial, right? There's a great deal that's already been done around EP and USP to be able to support certain methods and standardization thereof, but there are others, like frankly, biological activity where standardization is quite complex. So how do we actually drive towards that? So with that, just want to thank everyone for your time on behalf of Ramon and I. Really appreciate you sticking around, listening in. Hope this material was useful. And yeah, I see there are some questions already, so we'll look forward to diving right in.
Great, thank you so much to both Ezequiel and Ramon for a fantastic talk. We do have a few questions coming up in the Q&A, and as we allow people to start typing them in as well. Maybe I'll just start with one question. You both talked a lot about, this RUO and GMP and how important the transition is. But there's also this, constant kind of juggling between requirements, performance, and cost, right, during your drug development. So, could you also just share, your strategy on when to use which grade during your product development? And, Ezequiel, I think towards the end, you mentioned about qualifying a secondary vendor. And, I think the answer is, earlier is always better. But, could you just share your strategies around, when, if that's not feasible, right? When, what are the considerations to time the implementation during your product development?
Yeah, no, it's a great question, because if you go too early, you're going to be spending a lot more than you'd like, right? But if you go too late, you're going to be spending a lot later on, frankly, and doing a lot of work again. To me, there is a point, and this is probably something that needs to be discussed as early as preclinical, where prior to tech transferring an asset out of a university, I think it's really important to kind of have some of that work done up front, right? We're not expecting that university laboratories are going to be able to start sourcing GMP compliant materials to develop their therapies, but it should be a consideration when the tech transfer of an asset takes place, because ultimately you're going to have to pay your CDMO to do all this work to rework the entire BOM if you don't, right? And it's one of the hidden costs associated with tech transfer, right? You think you may be getting a fairly mature process, and at the end of the day, there's a ton of work that needs to be done to optimize it. You're always going to have to do work on the process as you move it out. But if you can mitigate the uncertainty associated with some of the variables, in particular the bill of materials, you're actually going to have a much more seamless translation. So I think part of it is kind of working with our partners in the academic setting to really consider this earlier. And I think beyond that, right, it should probably be one of the first things you do when you take on an asset. Really look at the bill of materials carefully, look at the process carefully, and really tear it down, look at all the constituent elements associated with risk. So I think it's never too early, but I do think it's obviously going to be complicated. But I would suggest, obviously, before filing your IND, having that work done, because if not, it's going to get quite complicated.
Great, thank you, Ezequiel. Let's see. So I've got some attendees questions. So for lot-to-lot consistency, does it need to be shown for the entire manufacturing process or for certain steps specifically? And how many batches are considered sufficient?
That's a great question as well. Typically, and there's two ways to think about it, right? Like your ancillary material developer will have their own assessment of lot-to-lot consistency, and it's going to be indicative rather than definitive, right? Ultimately, we will run a biological activity assay on our lots of serum or on our cytokines to really assess performance on our hand. But ultimately, these therapies are incredibly heterogeneous, and the cell types we're working with are incredibly heterogeneous. And so I think the best way to assess this is to, at the very least, take 3 lots of product, run them through a downscale model of your process from end to end, and see what the product consistency looks like, right? Holding all the variables constant where it's feasible, right? You may have to introduce some more complexity into your DOE to assess, different equipment or different dosing strategies around serum. But ultimately, looking at least 3 lots of certain proteins and serum, I think, can be helpful to establish some degree of statistical significance around comparability. But I think, again, we on the ancillary materials side can do only so much, right? We can assess biological activity, but ultimately it's going to depend on how it works in your process. And that can be a function, not just of the protein, but of the formulation, pH of the formulation, the excipients in the formulation. So there are a lot of variables that can lead to variability. And I think it's a matter of understanding them. And to understand them, I think you want to look at least a couple of different lots.
Great, thank you. All right, our next question, I guess to follow up on that, this is, I think, specific to one of the growth factors case study that you presented. What should be, according to you, the right way to assess bioactivity of a given growth factor if EC50 is not the right one?
Yeah, so I think it's going to depend on the growth factor and the cell line that's been established and is responsive to that growth factor, independent of other things. But ultimately, it's less about, to me, the analytical technique, as long as you're using a robust enough cell line to assess proliferation, or it could be gamma secretion. There's a variety of means that we measure. There are a variety of different ways of assessing biological activity depending on the protein. But I think it's more about how we analyze the data and establishing a robust means of looking at the sigmoid curves, understanding kind of the parallel line regions of these graphs to be able to move beyond a lot of the noise that exists at the extremities. So we use software for that, and happy to kind of share more information offline on really how we adopt that approach. And I think the other important consideration is always calibrating. And I know for GMP products especially, this is actually quite common in the field at this point to calibrate using an IBSC standard, of a known mass and activity, to be able to have some sort of baseline. Otherwise, we're relying on internal standards, which can make it complicated to look at several suppliers.
Yeah, and just to add, I don't think we're saying that EC50 is not the right one, but there's 3 factors here that we've noticed, obviously very impactful, and we need to be very cognizant of them. The mass as stipulated in the label of the product, that should always be checked, right? Sometimes there's discrepancies in that, or overdosing. Then obviously, like Ezequiel said, using internationally recognized, if possible, NIB standard. Otherwise, there are other NIB standards that may not be internationally recognized, but they're still very good standards to use. And then the cell line itself, right? You're going to have different EC50s basically based on the cell line that you utilize.
Mhm. Great, thank you both. I think we have time for a couple more questions. What approach would you use for designing qualification testing of bioactive ancillary materials like serum or cytokines? Do you use the assays on the certificate of analysis or develop custom assays to determine activity in your process?
Yeah, this is a great question, and it's always a complicated one. We don't expect it to be a core competence of a cell therapy developer to develop a biological activity assay for a cytokine. So what that means is you have to qualify a raw material without necessarily testing the core quality attribute that's going to determine how it performs in your process. So there's obviously, some degree of reliance on third parties there, right? So we can't expect that a cell therapy developer is going to bring in an HT2 cell line, develop a bioassay, validate that bioassay just to qualify interleukin-2, which might be one of 30 or 40 different raw materials they're using in their process, right? That's just not realistic. At the same time, you don't necessarily want to rely on a single vendor to establish that crosswalk or that raw material qualification for you either. So I think this is where we as a supply base need to do a lot more. We need to be able to establish, crosswalks between ourselves and our competitors and our collaborators in this space to provide off-the-shelf data packages that can help to supplement raw material qualification strategies on behalf of our vendors. And for serum, it's a slightly different approach, right? Serum is a much more complicated material to characterize and understand, and really we're assessing biological activity in terms of expansion with a given cell line, and that's only going to give us so much unless we go as far as mass spec and looking at each lot in a fairly sophisticated way. But I think it's important still to assess how that product is performing in your process. And I think that's probably, what us as an economist, I would say is the approach of satisficing, doing enough, doing what you can with what you have, right? And to me, that's going to be about testing materials in a small scale or downscale version of your process. And that's likely to be the best way to do things in the immediate term. But I think in the medium term, we need to get together as a supply base and create those crosswalks to be able to support colleagues.
Great. Okay, I think let's do. Oh, sorry, Ramon, do you have anything to add to that?
Well, I was just going to add that, we also rely on our own raw material suppliers, right? And not to add more fire to this, right? But I'm not sure if you guys are aware. I don't remember right now if it was the CTLO2s or the HT2s, but ATCC several years ago, maybe now, if I remember correctly, had sent out a message saying, hey, by the way, this cell line is supposed to come from Bob C mice, but we made a mistake and it's not. So again, just adding to that level of complexity, we can't turn over every stone, right? It will make it impossible and unsustainable. But yeah, it's just, we need to do as much due diligence as possible and try to standardize this across the board. And when we do see something that kind of like an outlier, well, then maybe we kind of go down that rabbit hole and really investigate further.
Mhm. Great. Okay, I have one last question here, and then we'll have to address everything else offline. So for processes that are heavily built on bill of materials containing biological complex materials during early development, what are your thoughts to move towards a more robust and more chemically defined process, considering the rush to clinical milestones and speed to market?
Yeah, again, it's a great point, and I think that's ultimately where we see a lot of this going. I think there are obviously challenges, right? Yeah, cells need, and oftentimes a source of human protein or animal protein to sustain growth and the expansion profile that we need to create a relevant dose. I think ultimately a lot of folks are pushing hard towards chemically defined materials in the BOM, and I think it's where we need to go, frankly. I don't think we're there yet, which is why our view has been, how can we de-risk the existing set of raw materials to a point where they can be useful, consistent, and applied in cell therapy manufacturing in the absence of a truly viable chemically defined approach, right? I know that there's a lot of work happening there, and in the future, you may well be there. But at this point, I just don't think we're there from a practical standpoint. However, the approach can either be, and they have to go hand in hand, right? Continue pushing towards more chemically defined raw materials, and at the same time de-risk, your recombinants, your human animal derived materials as well.
All right, I think we are at time right now. I want to thank you again, both Ramon and Ezequiel, for a fantastic presentation and for interactive Q&A. And thank you to the audience as well for participating in this webinar. Again, if your questions are not answered, we would address them offline and post them in the Q&A on our website. So thanks again, everyone, and have a great rest of your day.
Thank you, everyone.